On April 15, 2021, a US and Chinese team announced that for the first time, scientists had successfully grown embryos that included both human and monkey cells to a stage where organs began to form. The scientists were led by Professor Juan Carlos Espizua Belmonte of the Salk Institute in the US, who in 2017 helped make the first human-pig hybrid. This type of life forms are referred as chimera, named after the fire-breathing monster of Greek mythology that was part lion, part goat, and part snake. Whereas in modern medicine, chimera describes a living organism that contains cells or tissues with different genotypes. So, creating human-monkey hybrid, will it be a blessing or a curse? Before we start, please do subscribe my channel and press the bell icon to get my next videos. Human-monkey hybrids. In the human-monkey chimera experiment, a team of researchers implanted 25 human stem cells into embryos of crab-eating macaques, a type of monkey, then grew these embryos for 20 days in petri dishes before being destroyed in an attempt to investigate how the two types of cell develop alongside each other. After 15 days, the human stem cells had disappeared from most of the embryos, but at the end of the 20-day experiment, 3 embryos still contained human cells that had grown as part of the region of the embryo where they were embedded. Keeping human cells alive in the embryos of other animals for any length of time has proved to be extremely difficult. For scientists, the challenge now is to figure out how to maintain human cells in chimeric embryos for longer. What are the benefits of making chimeras? It is hoped that part human chimeras, essentially animal bodies with some human organs or other characteristics, might one day offer clues to help us treat human diseases, as well as providing organs to transplant to humans. Another expectation of this kind of research is the possibility of creating humanized animals that could serve as improved models for testing new drugs and boost the availability of tissue in biomedical research. Other so-called mixed species embryos or chimeras have been produced in the past with human cells implanted into sheep and pig embryos. If scientists can grow human cells in monkeys or other animals, it should be possible to grow human organs too. This would solve the problem of organ shortages around the world for people needing transplants. 17 people die each day waiting for an organ transplant. In the US, for example, more than 170,000 people, including men, women, and children, are currently waiting for an organ on the National Transplant Waiting List as of February 2021. 39,000 transplants were performed in 2020. The same researchers explored this avenue in pigs, seen as ideal because their organs are about the same size as ours. Also, their metabolism, for instance, diet and temperature, is close to the human metabolism. The key organs targeted in this research are the heart, liver, kidney, pancreas, lungs, and brains. Pigs would become essentially living containers for an unlimited supply of human organs. However, the actual proportion of human cells in the chimeras was quite low and their presence appeared to interfere with development, and the research failed. Monkeys are evolutionarily closer to us, so there is a greater chance that cells will interact effectively with each other. The stated goal of the human-monkey experiments is to understand and perfect the development of chimeras in primates before transferring the technology to pigs. Human-monkey hybrids might seem to be a much scarier prospect. However, their work could pave the way in addressing the severe shortage of transplantable organs as well as help understand more about early human development, disease progression, and aging. But for these purposes, part human chimeras will first have to be born, and this research takes us one step closer toward an important medical development than an ethical minefield. Ethical boundaries. Research involving the mixture of human and animal materials has been controversial from its inception due to the many dimensions of potential ethical conflict involved in this type of research. But how much of this work is ethically acceptable and where do the boundaries lie? Some scientists have raised concerns about the experiment, arguing that while the embryos in this case were destroyed at 20 days, others could try to take the work further. They also raised serious ethical dilemmas about the moral status of these post-human animals. Moral status is the concept of treating life forms according to their interests and capacities, such as consciousness, self-consciousness, moral capacities, and rationality. Chimera test subjects must be human enough to serve as effective models for health research, but not so human that they qualify for protection from this research altogether. A particular area of concern is the creation of chimeras with human brain cells. These organisms may be capable of self-awareness to the extent that they understand their identity and circumstances, which would produce unbearable suffering. These concerns about chimeric research add to the already potent ethical issues associated with mainstream invasive animal research. Tens of millions of animals are sick and injured, genetically manipulated, and killed in biomedical labs every year. Studies have shown that early cells of the central nervous system made from human embryonic stem cells can engraft and colonize the brain of a newborn mouse. This provides a proof of concept for possible cellular therapies. But what would be the ethical and cognitive status of a newborn rhesus monkey whose brain consists of predominantly human nerves? What safeguards do we need to ensure that the human cells don't also contribute to other organs of the host, such as the reproductive organs? However, chimeric research will and needs to continue. Many technical barriers have to be overcome before the production of human organs in pigs becomes a reality. In addition, some safety concerns must be addressed, such as the risk of zoonosis and the contamination of human organs by residual animal cells or animal proteins that could elicit potentially deleterious immune reactions in the organ receivers. Nevertheless, in view of the major medical or economic issues linked to this new technology, much research is currently focused on these issues. Rather than call for a ban or restricting funding for this area of medical research, it requires careful case-by-case consideration by independent oversight committees, fully aware of animal welfare considerations and recognizing existing standards. Unless we confront these issues now, we will find that our unrestricted efforts to save our bodies from sickness may come with an unwelcome cost of unforeseen consequences for which we are totally unprepared. What is your take on this? Do you support chimeric experiment for medical benefit, or are you against it on the ethical ground? Let me know your thoughts by commenting below. Thanks for watching. If you liked the video, please hit the like button, comment below, and be sure to subscribe Infoholic.